We're talking today about how genes are regulated. We're moving into biotechnology and molecular biology. So we were talking about, last time we were talking about transgenics and how you get organisms to express genes that you want. I want to start talking about more organismal. How do organisms and how do cells actually express the genes that they want to express? So we're going to talk about how promoters and how regulators work in terms of how the cell decides to turn on the proteins, the genes that it wants to turn on. So genes can be turned on or off depending on multiple different circumstances that the cell is in. So I'm really interested in development and how genes are turned on in the stages of embryology. So that's my first point. Genes can be turned on and off depending on the developmental stage that organism is in. Right? So at fertilization, the fertilized egg turns on certain genes. And as this embryo divides, you get to gastrulation, where the three main layers, the outer, the inner, the middle layer of the embryo is formed. Those different layers turn on different genes. At metamorphosis, at sexual maturity, at old age, you're constantly undergoing different gene expression. Your cells are in a different stage of life, and they end up expressing different genes. Now, once you're an adult, kind of your cell types have kind of been established, but even as adults and during the aging process and disease and menopause, I mean, there's lots of things that happen to an adult even that makes their genes express different, or makes their cells express different genes. Cell type. Now, this is in developmental systems when cells are actually deciding to be different from each other. So a kidney versus a liver, a neuron versus a skin cell. Different cell types are going to express different genes. The cells in your body are actually expressing different genes depending on what time of day it is. You have what is called a circadian clock. This is regulated mostly by your pineal gland, but your body knows actually what time of day it is, and it expresses genes on a regular daily cycle. So circadian means circa dia, about a day. So about the same time every day, you have different cells actually expressing different genes. Have you ever skipped a meal? Your body knows to anticipate that meal, right? You have a fairly regular cycle, and you don't get hungry necessarily because you're out of energy, right? You've got energy stores in your muscles, and you have energy in your liver, and you've got fat cells, and you can metabolize and go for several days without food, right? Your body has food and energy stored. But your body will start anticipating when you normally eat, right? Your body actually anticipates those, and it knows what time of day it is, and it knows this is the time of day you normally eat, and will start feeling hungry, right? So that's part of that is genes that are being turned on because of anticipating what time of day it is. So,
do your circadian rhythms depend on your sensory input?
Yeah. So you can retrain your clock to a different time zone, right?
Right.
When you have jet lag, you fly to a different time zone, and the sun is setting at a different time. The reason you can't sleep is because your body has the clock, and it knows what time it is. It knows what your time zone was, right? So it takes it several days, and most of this is input through your eyes, right? The lights that your eyes see will retrain your clock. So you'll have jet lag for a couple of days until your body acclimates to the new dark light cycle, and then you'll start being back on time schedule, right? You can kind of partially overcome jet lag by exposing yourself to light. You can anticipate the next cycle that you're going to be in, right? So if you're going to be off by a couple of hours, you can actually, by shining lots of fluorescent lights at yourself at the new time zone that you're anticipating, you can shift your body into going into that new time zone. Most of the time, that's not necessary, right? For whatever day and a half or so it takes you to readjust to the new cycle, it's probably not worth the 3 or 4 days that you'd have to gradually train yourself onto the new cycle. But you can do that. So it's dependent upon the light input from your eyes. Places where there's long, dark winters, they'll actually sit in front of fluorescent lamps for a good part of the day to try and keep their sleep cycle regular. Because if you're in the dark, up in the Arctic, and you don't see the sun for days and months, it's really hard. Your body doesn't know what to do, and so your sleep cycle and your eating cycle goes all out of whack. So you can partially make up for that by sitting under bright lights and training yourself to a cycle. So this brings in environmental conditions, right? To temperature, location, activity, injury. These are all going to change the expression of genes in your cells. You can actually force yourself to have different genes expressed as well. Psychologically, you can actually convince yourself of things. You can convince your body of things. You can actually change the gene's expression in your body, right? There's studies that have been done that actually self-talk, like giving yourself pep talks in the mirror, can actually change your chemistry of your brain. So you can partially overcome certain, and I'm going to be cautious to say this because this is, there are very real clinical depressions, and I'm not saying that it's completely your body or completely under your control whether or not you're depressed or not, because there are clinical conditions. But there's been studies that have shown if you have positive reinforcement, and even if that is self-talk, talking to yourself in the mirror, you can change the actual biochemistry of your brain and increase neurotransmitter expression, and the idea that laughter is a good medicine. If you just smile, you can force yourself in a sense to be happy, right? This is a fun thing to do. Force yourself to smile right now. You're going to feel stupid because you're like, what am I smiling at? But there's actually clinical studies where people who force themselves to smile all day actually report more positive emotions, and you can actually correlate that to higher expressions of neurotransmitters and stuff. You can force yourself to increase the neurotransmitters that your cells make just by smiling with a goofy grin on your face all day. It ends up making yourself laugh, right? If you just realize how ridiculous you are. All right, so let's talk the nitty-gritty of how how genes are actually turned on. I'll take the question, then we'll get into science here. So.
I was asking about placebos, but it is probably how it works.
Yeah. Placebos work because you've basically convinced yourself that you're going to get better.
Right, okay.
And there's nothing actually making you better, but that psychological effect is real, right? So we're going to start in a really simple system and a really simple gene expression platform here. We're going to talk about the choice that a bacteria makes between eating glucose and eating sucrose. What sugar is available in the environment dictates whether or not the bacteria will turn on the mechanism for just metabolizing glucose, or whether it turns on this additional apparatus for metabolizing sucrose. So, lactose. So we're talking about glucose or lactose, I'm sorry. So lactose sugar is a disaccharide. It's a glucose molecule and a galactose molecule bonded together typically between the 1 and the 4 carbons. Okay, so it's a disaccharide, two simple sugars put together in a disaccharide. Now, if there's high levels of glucose around, a bacteria will preferentially transport the glucose into their cell and metabolize the glucose. Right? It's easier to do. You can go right into glycolysis. You don't have to break the disaccharide bond and liberate the glucose in the galactose molecule. So if there's high levels of glucose around, a bacteria will just choose the glucose, and it will turn off the enzymes that are necessary for metabolizing the disaccharide. Okay? Now, if glucose levels are low, but there's lactose around, then the bacteria will say, well, I don't have glucose. That would be the easier thing to do. But what I'll have to do is turn on the genes that allow me to break down the lactose into glucose and galactose and put those through glycolysis. Okay? There's 3 genes that are necessary to do this. Number 1 is an enzyme called beta-galactosidase. The protein's name is beta-galactosidase. In the bacterial genome, the gene is called LAC-Z. Okay, so LAC-Z just means the genetic location on the genome. It codes for the protein called beta-galactosidase. Okay? What this does is it breaks the glycosidic bond that keeps those glucose and galactose molecules together. So it's a 1,4 glycosidic bond. This glucose and this galactose will be bonded together between those guys. The LAC-Z gene breaks that, separates the disaccharide into the 2 monosaccharides. You also need a gene called permease. That's the protein is called permease. The gene that codes for it is called LAC-Y. And this is a transporter. It basically says, the cell is saying, yes, I will bring in lactose into the cell. Normally it doesn't express that transporter in its membrane, and so it doesn't even bother to bring lactose inside the cell. Okay, so if you're going to choose to metabolize lactose, you have to make the transporter in the membrane to bring it in. The third gene is called transacetylase. It's coded for, so the protein is transacetylase. The gene location is called Lac A. This is an enzyme that's necessary for metabolizing the galactose, right? If you break a lactose molecule into glucose and galactose, glucose can go right into the glycolysis pathway. The galactose sugar, though, the transacetylase works on that to be able to get it to enter into the metabolism pathway. Okay, so galactose can't enter that first steps in glycolysis, so you have this additional enzyme, transacetylase, that kind of gets it into the metabolism pathway. So then I've been able to break down lactose. So in the bacterial genome, these three genes lie adjacent to each other in the genome. So this is the mRNA, but this is basically, right, just an RNA copy of the DNA of the genome. The LAC-Z gene that's going to code for the beta-galactosidase enzyme sits right here. Here's a start codon in the mRNA, and then there's a stop codon, and then there's this little intergenic region. But right next to it is another start codon, and the Lac-Y gene that makes that permease sits right next to it in the genome. This is just a couple of nucleotides. I don't know how long it is. I think there's a couple of dozen nucleotides that separate this. Okay. So the Lac-Y, the permease is coded for right there. There's a stop codon. There's a little intergenic region. There's another start codon. And here's the Lac-A, the transacetylase gene. All three of those proteins are coded for in this single mRNA. So when the cell recognizes I need to start metabolizing lactose, it makes a single mRNA, and that single mRNA codes for all three proteins. This is called a polycistronic gene. It means it has three coding regions for three different proteins. In this case, now poly just means any, so it could be a dicistronic, or you could actually have four. In this case, it's three. So three proteins get made off of the same mRNA. So all you have to do is get one genomic region turned on, and you get all three proteins necessary for metabolism. This is actually, so one RNA polymerase can sit down on the promoter region of this gene, and it makes this mRNA. And then one ribosome could sit down here. There's a 5' UTR region. That's where the ribosome sits down. The ribosome comes in, it makes the LAC-Z protein, so it makes a beta-galactosidase protein. And then it just slides along. It doesn't fall off at the stop codon. So the stop codon indicates I'm stopped making the beta-galactosidase. That protein gets released. The ribosome doesn't fall off, though. It slides over. It finds the new start codon, and that same ribosome makes the second protein. There's another stop codon. The permease is done being made. It slides over and makes the Lac-A. So the same ribosome makes all three proteins. Yeah.
What's the ribosome called? Do you know what the ribosome is called?
It's just the ribosome. Yeah, yeah.
So the Lac, is that the Lac Operon?
So yeah, this is called an operon. Right now I'm just talking about the RNA. So it's a polycistronic RNA. An operon means multiple genes under the same promoter, under the same regulation. So any polycistronic gene is by definition going to be part of an operon. Okay, so I'll get to that distinction in a minute. But you know, this mRNA, when this mRNA is made, that means all three proteins get made. Right? So they're under the same regulation. Any regulation that's turning on this RNA is turning on all three proteins. Okay. So Josh asked, is this an operon? An operon is anything that is where multiple proteins are under the same regulation. Okay. Now in a polycistronic, there's no way it couldn't be an operon, right? It's just the same mRNA. So all three proteins are regulated in the same way. Right? Whether or not I turn on this mRNA means I turn on all 3 genes. So here is the Lac Operon. Here is where that polycistronic gene would be made, right? LAC-Z, so this is in the genome now, right? This is the DNA. This is the LAC-Z gene, here's the LAC-Y, here's the LAC-A, okay? Now, there is a promoter region, and that's where the RNA polymerase actually gets recruited in, okay? So RNA polymerase sits down, the transcription factors recognize this green region of the genome, that's where RNA polymerase comes in. Now, adjacent to that is this little region called Lac-O. This isn't a gene in the traditional sense. This isn't a coding region. But what this is a regulator region. This determines whether or not the gene gets turned on or off. Okay? So it's called an operator here. If you have a mutation in this operator region, you will make these genes no matter what. This bacterial cell has a decision to make, right? It's got these genes in this genome, and it needs to recognize when it is appropriate to make these and when it is not. If there's plenty of glucose around, there is no reason to turn on these genes, because you'd preferentially just make glucose and not waste energy making these genes that aren't necessary. But if glucose levels are low and I need to start using a different sugar, then I should I should invest the energy in making this. So this operator is what's going to allow me to turn this region off and then turn it on when it's appropriate, okay? So this operator basically is a repressor. If that has the proper sequence, and if there's plenty of glucose around, this gene will be repressed and it will not be expressed, not be made. If there's mutations in the sequence in this operator, what ends up happening is RNA polymerase comes and sits down on that promoter and just starts making this. This is called constitutively active, always on, right? So if you have mutations in here that make that operator non-functional, I'll just make all this and I'll waste energy making enzymes that I don't need. Now, there's an additional gene. Yeah, Rachel, go ahead.
Are operators always repressors, or is it just this one?
I think just in this case. An operator is just kind of a technical term for something that's going to manipulate or is going to augment the expression levels. So let's keep it a generic term. It just means the thing that's allowing you to turn it on and off. Here, it's where a repressor is going to sit, okay? But you could think of it as a promoter too. I think operator is kind of a general term. Okay, yeah.
Okay, so it might be on that. I'm just having trouble understanding.
Sure.
So in this case, you're saying that it's specific that the operator is being repressed as to not make lactose? Or
Yeah, we're talking about turning this off, right? So this promoter region, a bacteria's RNA polymerase will sit down on that and make a gene, unless you specifically keep it from making it. Okay, so and that's where this is important. The sequence here is important for keeping RNA polymerase from sitting down on that promoter and making the downstream genes.
And so what you're saying is that in this case, it's acting as a repressor.
Yeah.
But in other
You could think of other scenarios where that is a place where it activates instead of represses.
Okay.
Yeah.
Okay.
Yeah. All right, so upstream of it is another gene. Okay. This gene has got a promoter in the green again, and it codes for the gene's name is called LAC-I. Okay. Just experimentally, geneticists found if there's mutations in the LAC-O, then all of these genes get made regardless of the environment. And if there's mutations in the LAC-I sequence, the downstream genes all get made as well. So this is a traditional gene in the traditional sense. A RNA polymerase sits down, I make an mRNA copy of this region, and that gets made into protein. Okay? So the way this thing works is normally where there's low levels of lactose and there's high levels of glucose, the bacteria wants to repress this. So in that case, RNA polymerase comes and sits down on this promoter. The LAC-I gets transcribed into an mRNA that gets translated into these proteins, these little arch-looking proteins in the illustration. These are repressor proteins. And what their function is they come in and they recognize the Lac O sequence. And those proteins bind to the DNA at the Lac O sequence region. And they basically keep RNA polymerase from being able to sit down on that promoter. So this is inhibiting these genes' expression, right? I have to make a protein, but that protein comes back and represses these other three. This is a traditional repressor protein. It's going to be a DNA binding protein, so it's going to have domains that allow it to bind to DNA. It binds in a sequence-specific manner, so it only binds when the proper sequence is there in the genome. So that's why if it's mutated, if you have mutations in the Lac O sequence, the repressor might not recognize the sequence and won't bind. And so then you'll get promoters coming in and making this. So you have to have a functional protein and a functional sequence that that protein recognizes to turn off this gene, to turn off these three proteins from being made. Yeah?
So the repressor that sits down on the Lac O
Yeah.
will stop it from...
Yeah, you just think of it as it's just sitting there and it's not allowing RNA polymerase to come and sit down. It's just like kind of taking up space, like not allowing RNA polymerase access to sitting down.
On the promoter region?
Yeah, so this guy is sitting down on the Lac O, and basically let's just think of it as just shutting the door on RNA polymerase being able to come in and sit down. Okay. Yeah.
So in low levels of lactose, it will land on that promoter, and in high levels, it will go on the other promoter? Or is
So this promoter is always on.
Okay.
Okay, there's no repression of that.
Yeah.
So no matter whether there's low levels of lactose or high levels of lactose, this gene, RNA polymerase comes and sits down on that promoter, makes the Lac I, and I make the repressor.
Okay.
Okay. So no matter what, the repressor protein is always made. Okay.
And how are you going to make, so you don't want to make Lac C or Lac Y? Or Lac
So yeah, the default state is to turn this gene off. Right? Default is make lots of the repressor. The repressor binds here and don't make this.
Okay.
Now a little bit squeaks through because that protein is binding and sometimes it doesn't bind. And there's a kind of a competition there between RNA polymerase and the transcription factors that are trying to get it there. So you get a little bit of making these proteins, but very, very little. Mostly you're turning it off. Okay? That's the default state. Default bacteria want to metabolize glucose and not lactose, so they shut this off. All right. This is called an inducible operon because what is going to induce it is the presence of lactose. Now, it's a little bit more complicated because also glucose levels need to be low, but I'm just going to talk about the presence of lactose, all right? If lactose levels go up, then the little bit of beta-galactosidase, so like I said before, a little bit of these proteins are being made, right? Even when repression is in its most robust state, I've got lots of that Lac repressor, still a little bit squeaks through, and I'm still making a little bit of beta-galactosidase and these other guys, okay? So what this does is it allows the bacteria to be sensitive to levels of lactose in the environment. If you would completely shut this off, you would have no permease, no lactose would ever get inside the cell. You would have no beta-galactosidase, and so just the bacteria would completely ignore it. But since there's a little bit of activity, there's a little bit of permease made, so it lets in a little bit of lactose if it's around. And there's a little bit of beta-galactosidase around, so if there's a little lactose that gets into the cell, I will break it down into glucose and galactose, okay? Now, if the lactose levels rise in the environment, some of that lactose will get into the cell. The beta-galactosidase will clip that, and occasionally it makes this molecule called allolactose. Basically, beta-galactosidase, its job is to break the disaccharide, so to break the lactose into glucose and galactose. Well, sometimes it does that, but then it sometimes sticks it back together in a different form. And this form, instead of being called lactose, is called allolactose. So it's a little side product. It's a byproduct of the process. So it's in very little amounts. But if you do make that allolactose, That allolactose, it's just a disaccharide, it's just a slightly different form of lactose, actually gets bound by the repressor protein. Okay? So the repressor protein binds a little bit of this, and that will inhibit those repressors from being able to go up and bind to the Lac O. So a little bit of lactose basically takes out of the equation a little bit of the repressors. If lactose levels continue to rise up, then I inhibit more and more of the repressors, right? It's kind of a thermostat in a way, right? If lactose levels rise, then some of that beta-galactosidase makes the allolactose and inhibits some of the inhibitors, which means I make slightly more of this because it's not able to repress as well as it used to. Okay? Now, if lactose levels continue to rise, then I will continue to repress more and more of those repressor molecules, right? I'm repressing the repressor. So in contrast to lactose levels rising, so will the expression of beta-galactosidase in these genes. Those will rise as well. And if the glucose levels are low and lactose levels continue to rise, I will start repressing more and more of those guys, making more and more of this, and I then switch over and start metabolizing the lactose instead of the glucose, using the sugar that's available. Yeah.
So it doesn't sound like, what I thought before was that the cells would recognize, okay, low levels of glucose, high levels of lactose, let's switch over to making lactose,
Yeah.
or breaking down lactose. It sounds like just in the presence of lactose, does that mean if there's high presence of lactose that there's necessarily low presence of glucose?
Yeah, I'm saying let's take glucose out of the equation. Say that there's no glucose around,
Oh, okay.
and if there is lactose around, then it will start to do this.
Okay.
So the issue is actually a little bit more complex. If there's lots of lactose around and lots of glucose around, yeah, there's additional regulation that the bacteria undergoes, and I don't want to get into that. So we'll just assume glucose level is low, and that it's going to select for using this sugar instead. Yeah.
Did you say that allolactose is just one of the byproducts of?
It's basically kind of a mistake that beta-galactosidase makes. So beta-galactosidase, its job is to break apart the lactose molecule into a glucose and a galactose, right? Well, sometimes it doesn't do that. It breaks them apart, but then it sticks them back together in a little different conformation. And so allolactose is just an alternate version of glucose and galactose being together. So it's like a side byproduct.
You have to be having a lot of lactose breaking them apart
Yeah.
to get enough of.
Yeah, so initially very, very few molecules actually get made into this allolactose form. Yeah. So you have to have a lot of lactose metabolism going on before you repress this in any sufficient amounts, right? So does this make sense, how you repress the repressor? If you repress the repressor, that means nothing binds to the Lac O. RNA polymerase is free then to come sit down on this, and it'll make everything.
What's free? Sorry.
Well, to get this to be made, we need to make this into an mRNA first. And so you have to have RNA polymerase come sit down on the green region, the promoter region. And if it's allowed to, if there's no repressor binding to the Lac O, then RNA polymerase will sit down, and it'll move along, and then it'll actually start, making an mRNA at the transcriptional start site, right? So. Most of the time, bacteria are haploid, right? They only have one copy of the genome, right? So in normal cases, basically they're just working with this, right? And this is the normal scenario, right? To figure out how this is working, what people did is introduced different variant versions of the Lac Operon on like a plasmid and made the bacteria heterozygous for these sites to try and figure out how this is working, okay? So here is a mutant form of the E. coli chromosome, the genome, right? So it's got a functional Lac I It's got a normal sequence of Lac O. It has a mutant copy of Lac Z. Basically, we're just thinking of this as a reporter. What kind of thing is being made? So we're basically setting up a competition between these two. We're making it a heterozygous state. They've got one version and they've got a different version. The way we know that genes are being made off of the genome is whether or not they make a functional Lac Z. The second copy that we put in on a plasmid, so here's an F factor plasmid. On this plasmid, we stuck another copy of the Lac Operon, all of these genes here. In this case, this is mutated at the Lac O. I've got a faulty mutated sequence in the Lac O. The readout for this is we'll make a normal Lac Z gene, but I'll make a mutant form of the Lac Y gene. I just want you to basically think of these as reporters, right? Which chromosome is being made? If the chromosome is being, or if the transcript is being made off of the genome, it's going to have a bad version of Lac Z. If it's being made off of the plasmid, it's got a bad form of the Lac Y. So just think of those as readouts of which one is being made, okay? So you set this up and you ask the question, what's going to happen as these two interact with each other? What ends up happening is the Lac O is the important part of this equation right now, okay? Because I'm making a functional repressor. Both of the operons are making the functional repressor, okay? Here, so the repressor then is going to be able, since this is a protein, the protein is made, the protein circulates around in the cell, finds access to the sequence, right? So this is acting in what we call a trans manner. The protein acts in trans because it can act on its own repressor, or it could trans and act on the other chromosome. Okay? So trans means it's soluble, it's in the cytoplasm, it has free access to either chromosome. Okay? So it will influence its effect on either one. The Lac O, its influence is called cis, because its influence is on whatever is adjacent to it in the chromosome. This is just a DNA sequence, Lac O is, so there's no way that it can go have a function over on the other chromosome. Okay? So if I have a mutant form of this, then the repressor is unable to bind. It doesn't matter whether lactose is around or not. The repressor can't bind to it, and so I get a constitutively active mRNA off of this chromosome, right? And so the readout tells me, oh, I'm making this mutant form of the Lac Y, so therefore this must be the mRNA that's being made. The actual chromosome is still repressed. It has a functional Lac O, so the repressor comes. This acts in a cis manner, so it influences what's directly adjacent to it in the genome, and this thing gets repressed. Okay? So all I'm trying to get you to see is what the difference between a trans-acting factor and a cis-acting factor is. Okay? Cis means its influence is directly related to what it's next to. So if I have a functional Lac O, I do the proper function on my adjacent. If I have a non-functional Lac O, then it has its influence just on the sequence that it's adjacent to. A trans-acting factor is going to have equal influence on both chromosomes. Right?
So basically, if you make repressor, I don't know, proteins from the LAC-I on one chromosome, it can't affect the other chromosome?
It can't affect the other chromosome.
Oh, it can? Okay.
No, cis. A cis-acting element means it only has its influence on what is sitting physically next to in the genome. Right? So this LAC-O is a cis effector.
Okay.
It only affects things that are adjacent to it. The function of this LAC-O, whether it's good or whether it's bad, makes its influence on what it's directly next to. Okay. So typically, and I'm going to show you variations now when we get later, but typically a sequence will be cis-acting. If this is just a DNA sequence, then it's only going to influence what it's next to. Okay. A trans-acting element is going to be typically a protein that gets made and gets to diffuse around the cell, and so it can act on either one, right? It can exert its influence. Now, it's unable to touch this one, right? But it actually has the possibility of getting over there, right?
So LAC-I is trans, LAC-O is cis.
Yes, so LAC-I is trans, LAC-O is cis, cis-acting elements. Yeah.
So I'm just trying to understand cis and trans. So trans, if the LAC-O was trans, does that mean that the LAC-I from the top one could influence the LAC-O from the bottom?
If LAC-O was acting in a trans manner, then its mutant dysfunctional form here would make this get made. So what this guy's role is to normally repress, right? The repressor comes and it binds to it and would turn this off, right? Because this is mutated, I now make this, right? I'm making the mRNA, that gets made into protein, and I see, oh, I'm making something off of that chromosome, right? That mutation, though, only acts on what it's adjacent to. It has no influence on the other chromosome in the cell, because the other chromosome in the cell is regulated by the LAC-O, right?
Oh, got it.
So it's acting in cis because that is only exerting its influence here. The mutant form does not have any effect on the other guy. He's got his own cis element that regulates him.
So if you did see both get made, and knowing that the LAC-O on the top was cis, you'd assume that the LAC-O on the bottom was trans then?
Correct, right.
Okay.
If by introducing a single mutant copy, it got this made and it also got that made, I would know there's some way it's influencing the other chromosome. Whether it's cis or trans is based on the readout, right? This is getting made, so I know that that mutant is having its effect on this version. This is not being made, this is getting repressed, so I know that this guy does not act in a trans manner. Even though it's mutated, it has no effect on the other chromosome, right? Now, you can think of a reverse situation. If this guy is mutated, then the trans effect could have an effect on both sites. Which I was planning to go into next time, because I thought we would spend more time on this. I don't have the slide prepared. Yeah.
So earlier on the first Lac Operon slide, it said a mutation in LAC-O causes like an increase in the LAC Z, Y, and A proteins. Is that because it does not allow the repressor to sit on it? That's the reason why there's an increase.
Right. So if you mutate that sequence, then the repressor that's being made from this doesn't recognize that it should sit down there. If it doesn't sit down, RNA polymerase comes in and all those just get turned on, right? The take-home message, RNA polymerase is dumb, right? RNA polymerase will just sit down if something's available in this case. Now it's a little bit more complex than that, but in this case, just think of like, if that site is available, RNA polymerase will sit on it, right? And what's controlling this gene's expression is whether or not a regulator comes in and allows it to sit there or does not allow it to sit there, right?
Why did you have, remember, E. coli or haploid?
Well, because normally you only have a LAC-O, right? So you only have one chromosome in E. coli, right? So a normal E. coli have one LAC-O, and that acts in a cis manner.
Okay.
The LAC-I, though, we don't know whether that's acting in a trans or a cis manner, unless you have a second chromosome for it to act on, right?
Gotcha.
So in order to determine whether things are acting in a cis or a trans manner, you have to have two chromosomes and decide who's acting on which one, right? So normally this isn't an issue for a bacteria, right? He's got its LAC-O and its LAC-I, and he's going to be influenced by mutations in either one, right? But if it's haploid, then we can say, well, what if there's a good copy here and a bad copy here, and we can do mixing matching experiments, right? So this is where it's handy in bacteria. You can actually get them to be diploid at sites if you want them to, because you can put in a plasmid and give them a second copy of a gene and see how the two versions interact. So bacteria is kind of a fun little playground for you to see how genes interact, right? Yeah.
So just a general question on how these affect the person, or does this work the same way in human body?
No, so it's going to get more complex in humans and in eukaryotic cells. So bacteria are like the simplest version. This is like the simplest example of how a gene can get turned on and off by an environmental input, right? So I've just said there's all these environmental inputs, right? Developmental stage and the age of the cell and whether it's day or night is going to determine how genes get turned on and off. This is just like the simplest, most straightforward way of like, how does a gene get turned on and off? Now, the same kind of principles are going to hold true, but we're basically going to have to add layer upon layer of regulation when we get to eukaryotes. Basically, a bacteria says, all I'm really interested in is turning on an mRNA. If you make an mRNA, then that mRNA is going to be turned into protein, and it's going to be expressed, right? So the level of control in a bacteria is whether RNA polymerase sits down and makes your mRNA. If you get that step going, you're going to express the protein, and that protein is going to express its function, and that cell is going to change based on whatever that protein's doing. In the eukaryotes, we're going to have layer upon layer of making sure you get the proteins doing their function at the right time and place. So, yeah.
Are we capable of do we have, like you were saying, that they'll break down glucose just naturally, that's what they want to do. But if they need to, they can break down the lactose if it's present, if that's all that's present.
Right.
Do you have that ability to do that too? Like, I'm thinking of just someone who's like lactose intolerant, who can't break down.
Yeah, I mean, your liver is famous for doing this, right? Whatever your liver is presented with, basically, it will change its gene expression to deal with whatever that is. So if you, for instance, I know the first thing that jumps to my mind is consuming alcohol, right? If you consume alcohol, then you've got alcohol in your bloodstream. Your liver then turns on enzymes necessary for converting alcohol into fats and breaking it down, right? So, I mean, your pancreas cells do this, your kidney cells do this. I mean, all of your cells, depending on what they need to do, will increase protein expression to do that new function that it needs to do. And then if alcohol goes from your system, most of the liver cells are going to turn off those enzymes necessary for metabolizing alcohol, because we don't need to do that anymore. So quit spending the energy to make proteins that you don't need. So you shut off that protein expression and wait until you need it. So, yeah. Now, your liver is doing all this amazing metabolism depending on what's flowing around your bloodstream. So there's a lot of diverse gene expression going on depending on what you just consumed and what your liver's trying to detoxify and things like that. So, yeah.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.